Okay, today we're going to be talking about signaling pathways. More importantly, we're going to be talking about the GPCR, the G protein coupled receptor signaling pathway. All right, signaling pathway here. So let's get into it. So first of all, let's take a look at this diagram over here. Okay, so what we see is we have a ligand and then we have a receptor. The ligand will bind to this G protein coupled receptor. When that happens, the G protein coupled receptor undergoes a conformational change, meaning its shape changes a little bit. And that is very important because what we see here is it's called a G protein coupled receptor because in the inactive state, we have this G protein right here. The G protein has an alpha subunit and it has a beta gamma subunit, just like that. As we can see, when inactive, it's just bound there, it's right there next to that G protein coupled receptor. When the ligand binds, we have a conformational change in the receptor. It causes this G protein right here, which has GDP bound at rest, it causes the GDP to be released and it binds GTP. So it goes from GDP to GTP, causing it to be in its active state. When this occurs, this beta gamma subunit and this alpha subunit will separate. They'll go their two separate ways, okay? The beta gamma subunit will hang around, kind of around where it's at. It's not lipid linked, it's not anchored into that membrane, anything like that, but it'll hang around waiting for the G protein to come back, or the alpha subunit to come back to it. So what happens with this alpha subunit? Let's erase these arrows right here. The alpha subunit will go and it will interact, as we can see here, with this enzyme. And what is that enzyme? It's called adenylyl cyclase. So alpha subunit comes over here, interacts with adenylyl cyclase, and then adenylyl cyclase is important because it will make what we call CAMP. It's cyclic AMP, and that is our secondary messenger. A secondary messenger essentially is what helps a signaling pathway to continue on to cause a kinase cascade or to cause whatever its desired effect is downstream. It helps cause the downstream effect, okay? So cyclic AMP is our secondary messenger that is made from this enzyme adenylyl cyclase, which is activated by the alpha subunit. So what does the cyclic AMP do? Why is it important? As we can see in this illustration, we've got, what is this? We'll talk about it. We've got our CAMP here, and it's interacting with whatever this is. So what is that? This is our pKa. So we'll come down here and look at this. pKa, protein kinase A. And what we notice here is we have 4 different subunits. So 2 of them are similar to each other, and the other 2 are similar to each other. And the C stands for catalytic, and the R stands for regulatory. At rest, the catalytic and regulatory subunits are bound together. When cyclic AMP comes into the picture, it will bind. So 2 cyclic AMP, as we can see here, will bind to each regulatory subunit, and it releases these 2 catalytic subunits, okay? And then these catalytic subunits, they can go and phosphorylate. They can go phosphorylate their target proteins and cause further effect downstream, okay? So we're not going to get into that a ton, but just know, we'll go over the basics one more time. Ligand binds to G protein receptor, G protein coupled receptor. The inactive form of the G protein releases GDP, and it attaches GTP, it binds GTP. Alpha subunit detaches from beta gamma, it goes and it activates the adenylyl cyclase enzyme, which will then make our cyclic AMP our secondary messenger. Our cyclic AMP secondary messenger will bind to the inactive form of pKa. It will bind once again to CAMP to each regulatory subunit, releasing these two catalytic subunits with their serine and threonine residues to go and cause further kinase cascade downstream. And that's all we'll get into today about the GPCR signaling pathway. And these pictures are coming from our book, from this source. Thanks for tuning in.